Structure with the unwound parental duplex forming the tail of the fork. This replication fork forms at the replication origin, where it employs the various proteins required to initiate the replication process, and subsequently moves along the template DNA strand. This movement can either be in a single direction from the origin till the replication terminus, the point in DNA where the replication process ends, and the phenomenon is called unidirectional replication, or it can be bidirectional, where two replication forks proceed in opposite directions from the origin and converge at the terminus. DNA replication is semi-conservative in every round of DNA replication. Effectively, two new daughter strands are formed from the two parental DNA strands of a DNA duplex. Theoretically, this can arise two possibilities in a conservative mode of replication. Two daughter strands that are formed from the parental templates would intertwine and form a brand new DNA duplex themselves, or else, in a semi-conservative mode, parental DNA strands would separate permanently, each forming a DNA duplex with its complementary daughter strand. While the conservative mode is seen elsewhere in the biological world, it is not seen in the case of DNA replication. Figure 3.1. 3. Molecular biology. DNA replication. DNA replication is the event that marks the beginning of cell division. When a cell prepares to divide, its entire genomic DNA sequence undergoes a round of duplication in a complex mechanism employing several proteins and resulting in a new DNA duplex which is eventually sent off to the daughter cell after each cycle of cell division. The unit of replication is called replicon. A replicon is usually a DNA molecule where replication occurs. Since prokaryotes contain a single DNA duplex, they usually have single replicons. While eukaryotes, having more than one DNA strand, have multiple replicons. A replicon contains one origin of replication. As the name suggests, an origin is a site along the DNA molecule from where the event of replication begins. It is a cis-acting element since it affects only the replication of the DNA strand on which it resides. While the sequence of origin may vary among different organisms, mostly it is an at-rich region that aids in the unwinding of the parental DNA helix to make room for the formation of each new daughter DNA strand along with the parental templates. This point at which the parental DNA duplex unwinds and opens into the two newly formed parent daughter DNA duplexes is called a replication fork, as it resembles a fork-like. Figure 3.13 models of DNA replication. In the conservative model, parental DNA strands blue remained associated in one DNA molecule while new daughter strands red remained associated in newly formed DNA molecules. In the semi-conservative model, parental strands separated and directed the synthesis of a daughter strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. In the dispersive model, all resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA. In 1957, Mendelssohn installed M&S showed in their famous autoradiography experiment, figure 3.2, that DNA replication always occurs in a semi-conservative mode where each new DNA retains a parental DNA template strand. The basic idea is to use density as a label to distinguish all the new pieces of DNA. M&S grew bacteria for a long time in a medium containing heavy N, 15N, so all the DNA was heavy. Then they grew the bacteria for one generation in a medium containing lighter ordinary N, 14N, so all new DNA strands or newly made sections should be light. Then they extracted the DNA from the cells and measured the density of the DNA. All the DNA was of a hybrid density between heavy and light. This result is consistent with semi-conservative replication and dispersive replication, and it rules out conservative replication. If replication were conservative, then after one generation, one half of the DNA, the old molecules should be heavy, and one half the new molecules should be light. M&S grew the bacteria as above in a medium containing heavy N, and then grew it for two generations instead of one in a medium containing light, and then they looked at the density of the resulting DNA, what you get after two rounds of DNA replication. The result is in the text, and you should be sure you understand how the result rules out dispersive and conservative replication. M&S did another experiment to rule out dispersive replication. They took the hybrid DNA obtained after one generation of growth in light and denatured it. They reasoned that if replication is semi-conservative, the denatured DNA should be one half heavy and one half light, or one half old strands and one half new ones. If replication is dispersive, each individual single strand should be part heavy and part light, and so all the denatured DNA should be hybrid in density. The denatured DNA was one half heavy and one half light, ruling out dispersive replication and supporting semi-conservative. Figure 3.2 Mendelssohn installed M&S experiment. They showed their famous autoradiography experiment to prove that DNA replication occurs through semi-conservative mode. Theta replication, Q replication, in E. coli and other prokaryotes, DNA is usually circular. Therefore, when replication in these types of circular DNA occurs, the molecules assume the shape of the Greek letter Q, and this mode of replication is thus called Q replication. A typical illustration of circular DNA replication is shown in figure 3.3. The DNA of plasmids, phages, and viruses are seen to be circular. In eukaryotes, mitochondria mtDNA and chloroplast cpDNA contain circular DNA. Q-type replication is also found in many E. coli plasmids such as col1, PSC101, R6K, F plasmids, etc. Figure 3.3 Circular DNA replication via Q intermediate. Circular DNA has a single origin of replication from where two replication forks originate and proceed in opposite directions. The forks expand as new daughter strands are extended during replication, as shown. The first evidence of circular DNA of E. coli came as early as 1963 from J. Cairns' autoradiography experiment. In this experiment, E. coli cells were fed with radioactive 3H thymine so that all newly synthesized DNA would be radioactive. After isolating and storing the DNA for months, when the 3H atoms in the DNA molecule decayed, they emitted electrons which were visualized as dark rings on an autoradiograph. The pattern of the dark rings eventually took the classic circular shape resembling Q replicating a circle. Figure 3.4. Daughter duplexes. Replication fork parental duplex. Replication fork. Figure 3.4 autoradiograph of a replicating chromosome in E. coli. Cells have incorporated radioactive 3H thymine in their DNA for two generations, which are represented by the continuous lines of dark rings in the photographic emulsion. The results of the autoradiography experiment also suggested that replication can be bidirectional. Whether replication is unidirectional or bidirectional depends on whether the replication forks move from the origin in both directions or only one direction. This could be visualized in electron micrographs where the replication bubble would expand in both directions in case of bidirectional replication. Although bidirectional replication is most common in prokaryotes, it is also seen in eukaryotes like yeast, fruit fly, and mammals, while unidirectional replication is seen in mitochondrial DNA replication in invertebrates. An illustration of bidirectional replication is given in figure 3.5. In bidirectional replication, a replication bubble is formed at the origin from where two replication forks proceed in both directions. Each replication fork exhibits leading and lagging strand synthesis simultaneously. No end problem in B. DNA replication. Overview of enzymes. The underlying principle mechanism of replication is the synthesis of new DNA strands by the addition of DNTPs. Figure 3.6 forming phosphodiester bonds between the 3' oxygen of the growing strand with the A phosphates of the DNTPs. Even though DNA polymerase performs this function of chain elongation, it itself is unable to initiate the process de novo. To carry out one event of replication, various enzymes are recruited by the cell, which play pivotal roles in the process. Hence, it is imperative to understand the roles that each enzyme plays in the process in a chronological order to effectively grasp the overall mechanism of replication. Figure 3.5 Mechanism of bidirectional DNA replication. Figure 3.6 Structure of DNTP showing A, B, G phosphates. Topoisomerase. Most naturally occurring circular DNA are negatively supercoiled, meaning they have a linking deficit which causes them to have fewer turns in the duplex than would be found in nectar linear DNA of the same length. However, during replication, unwinding in the replicating region introduces positive supercoiling in the adjacent unreplicated region of the template strands. As a result, the replication fork advances, the positive supercoiling increases, and the unreplicated DNA becomes more and more tightly coiled. Such tightly coiled DNA would be impossible to unwind after a certain time for the replication to advance in this region. For this reason, the action of a particular enzyme comes to play. This is known as DNA topoisomerase, which introduces negative superhelicity. All organisms contain one or more topoisomerases. E. coli has 4 types of topoisomerases IID. These topoisomerases maintain a particular superhelicity of the DNA helix in vivo, which was proved by the experiment conducted by Goldstein and Ehrlich in 1984. Usually, lower temperature increases the superhelicity of DNA. They observed that bacterial DNA extracted from cells grown at lower temperatures exhibited fewer helical turns than expected. From this observation, they assumed that cells maintain a constant superhelicity of DNA that balances off the effect of environmental or other factors. At the start of replication, the supercoiled DNA is converted to relaxed state by topoisomerase I. Figure 3.7. This enzyme breaks the phosphodiester bonds in one template strand and passes it through the other, creating a localized uncoiling at the replication site. This mechanism does not require ATP hydrolysis as the enzyme uses energy from the supercoiled DNA strand. As discussed earlier, during replication, movement of the replication fork along the template strand creates positive supercoils in the adjoining unreplicated region of parental DNA duplex ahead of the replication fork, which in turn creates a huge amount of torsional stress. This stress is released by the introduction of negative supercoils by topoisomerase 2 or DNA gyrase, as a result of which DNA returns to its original condensed state after replication. E. coli DNA gyrase consists of 4 subunits 2A and 2B subunits. A subunit nick will hydrolyze both the DNA strands and pass one strand around another part of the helix, thus introducing a supercoil and eventually sealing the nicks. These subunits have APase activity which provides the energy required for nicking and sealing processes. Figure 3.7 topoisomerase I introduces negative supercoil and reduces torsional stress on DNA. 1. Electron micrograph of the DNA of SV40 virus. When the viral circular DNA is isolated in vitro and is devoid of its associated protein, the DNA adopts a supercoil configuration. 2. Electron micrograph of the DNA of SV40 virus. When the supercoiled DNA is cleaved on one strand and DNA assumes a relaxed circular configuration owing to the loss of supercoil. This reaction is accomplished by the activity of topoisomerase I, which catalyzes the cleavage on one strand and resealing of the broken ends. Helicase. The parental strands of the replication origin unwind by a process called melting and is executed by the enzyme helicase. The helicase activity is present in both prokaryotes and eukaryotes, and the corresponding names of the proteins are listed in table 3.1. DNA helicases and rep protein take part in this unwinding of the duplex in an ATP-driven mechanism and break the hydrogen bonds holding the template strands. E. coli has 12 different helicases, of which DNAB is a major unwinding helicase. Once separated, the single-stranded binding proteins SSB specifically bind to the single-stranded DNA and prevent reannealing or damage of the strands. Primase. Once the strands are separated, a specialized RNA polymerase or primase synthesizes a short stretch of RNA, usually approximately 10 to 12 bases, complementary to the unwound template strands. This RNA fragment acts as a primer for the subsequent DNA polymerase activity and binds to the template strands due to its complementarity. Primase in E. coli is a product of the DNA G gene. Primase is activated by DNAB to initiate the elongation of the leading strand and Okazaki fragments of the lagging strand. DNA polymerase. There are several types of DNA polymerases in E. coli. The main replication enzyme is a multi-suited protein, DNA polymerase III, which is the main catalytic part of the DNA polymerase III holoenzyme. DNA polymerase I and II are single polypeptide enzymes. DNA polymerase I repair DNA damage, and DNA polymerase II is responsible for restarting replication once it is blocked due to DNA damage. DNA polymerases IB and B are called error-prone polymerases, responsible for continuing replication by bypassing the DNA damage. DNA coli is involved during the initiation of replication for the removal of RNA primers and replacing them with short stretches of DNA. The 5' 3' exonuclease activity of polymerase I executes this function of removing the RNA-DNA linkage, which is otherwise resistant to RNase H activity. However, because of the low processivity of DNA polymerase I, it cannot add more than 20 to 100 nucleotides. This process of making the DNA at one point and extending it for a stretch of nucleotides so that the nick translates along with the DNA is called nick translation, figure 3.8. This process is utilized greatly for the radioactive labeling of DNA. Figure 3.8 nick translation by DNA polymerase I. The 5' 3' exonuclease activity of DNA polymerase I green cleaves off nucleotides from the 5' end of the nick. The 5' 3' polymerase activity then adds nucleotides to synthesize new DNA and translates the nick to a new position in the 3' direction. The removed nucleotides are labeled in blue. The parent strand is labeled in black, while the new DNA is labeled in red. Since the main event of replication involves both polymerases I and III, both the enzymes show high accuracy. Therefore, both DNA polymerase I and III have 3' 5' exonuclease activity, i.e., they can remove a nucleotide from the 3' ends of the DNA if it is incorrectly incorporated. This is known as the proofreading function of polymerases. Clonal fragment. DNA polymerase I is a 109 kD protein that can be cleaved into two parts by proteolysis. The larger part, 68 kD, is known as the clonal fragment, which has both polymerase and a 3' 5' exonuclease function, figure 3.9. The smaller part exhibits 5' 3' exonuclease activity. The latter function makes DNA polymerase I a unique enzyme that can conduct the nick translation of DNA during replication. Figure 3.9 different functional domains of DNA polymerase I. Leading strand takes place in the direction in which the fork moves, but the problem arises in the case of the lagging strand synthesis, which urges the need for polymerization to take place in the opposite direction of the replication fork. Since it becomes an impossible situation for DNA polymerase III to elongate the lagging strand, alternatively, a new primer binds to the template for the lagging strand in a 100 to 200 nucleotide interval, each of which is then elongated in the 5' 3' direction by DNA polymerase III till the beginning of the next primer. This results in discontinuous segments of the daughter strand, which are termed Okazaki fragments, after the name of the discoverer, figure 3.10. The RNA primers are removed and replaced by DNA elongated from the previous Okazaki fragment in a process called nick translation carried out by DNA polymerase I. The discontinuous DNA fragments are eventually joined by DNA ligase. DNA polymerase I was first discovered by Arthur Kornberg in 1958. The 109 kDa polypeptide has three functional domains. The 5' 3' The 5' 3' exonuclease domain functions to remove nucleotides from the 3'. The 5' 3' exonuclease domain functions to remove nucleotides from the 3' end of an ssDNA. This role is important in the removal of RNA primers during replication. The 3' 5' exonuclease functions in proofreading nucleotides and removing any misfire base from the 3' end of the grown DNA strand. The 5' 3' polymerase activity fills in the gaps in DNA by adding nucleotides. Proteolytic cleavage results in a larger C-terminal fragment that retains the 3' 5' exonuclease and polymerase activity and is known as the clonal fragment after the name of the discoverer. Clono is widely used for practical purposes like the synthesis of radioactive probes. The problem with DNA polymerase activity is that it carries out chain elongation only in the 5' 3' direction. Hence, chain elongation for the figure 3.10 illustration of a typical replication fork. The lagging strand synthesis is discontinuous as it proceeds by sequential synthesis of Okazaki fragments. Synthesis of each fragment begins by loading a primase indicated by hatched lines and synthesis of RNA primers indicated by blue lines. The leading strand synthesis takes place continuously in 5' 3' direction. The black arrow indicates the direction of the fork movement. The template strands are indicated by solid black lines. Daughter strands are indicated by solid red lines. Figure 3.11 mechanism of DNA ligase reaction to seal the DNA ends. Okazaki fragment. Raiji Okazaki in 1967 reported that when E. coli cells were fed with radioactively labeled thymidine for a very short time, few seconds, and killed instantly, the size of the radioactive DNA extracted from the cells was found to be very small, till 1,000-2,000 nucleotides in sucrose density gradient profiles. On the contrary, when cells were fed with a radioactive base for a longer time, 1 to 2 minutes, the size of the extracted radioactive DNA fragments appeared to be much larger in sucrose density gradient profiles, figure 3.12. This suggested the synthesis of very short fragments in the initial stage of replication when part of the DNA is synthesized discontinuously, which went on to form larger fragments with time. These short fragments are later named Okazaki fragments after the name of the discoverer. Figure 3.12 experiment demonstrating the synthesis of Okazaki fragments. Profiles of sucrose density gradient centrifugation of radioactive binding labeled DNA of phage-infected E. coli cells is performed by R. Okazaki et al. in 1968. When cells were treated with short pulses of radioactivity, a large amount of radioactivity was detected in very short fragments of DNA, observed by sucrose density gradient centrifugation. With increasing the time period of pulse, the radioactive fragments appeared to be lengthier. This explained the synthesis of very short DNA fragments in the initial stage of replication, which later on became ligated, forming high molecular weight molecules. DNA polymerase III is a large multimeric complex of 900 kDa with 10 different types of proteins organized into 4 subcomplexes. One, the catalytic core complex with 2 copies of core polymerases, each of which is employed for leading and lagging strand synthesis. The core polymerase has 3 subunits A, E, and Q. While pol solely carries out the polymerase function, pol E and Q are responsible for exonuclease activity. 2 copies of T subunit which is responsible for dimerizing the core subunits together. 3 2 copies of the clamp, each consisting of a dimer B subunit forming the B ring, which tethers the core polymerases onto template strands and ensures process of T. 4 the G complex or the clamp loader, comprising of 7 proteins. This complex helps load the B clamp on the DNA. A simplified structure of DNA polymerase III is illustrated in figure 3.13 and figure 3.14. Figure 3.13 different subunits of holoenzyme DNA polymerase III. The core polymerase consists of A, E, and Q subunits. B subunits comprise the clamp, and G complex forms the clamp loader. G complex consists of G, Y, C, D, D, and T subunits. Figure 3.14 model of pol-free binding to DNA. Sliding clamp B, an exonuclease 3A surface representation of polymerase and DNA, with the C-terminal region of pol-free and sliding clamp B indicated schematically, is shown B interaction sites of exonuclease 3 binding to pol-free, with 3 binding regions in pol-free, Bjork and McHenry, 2006 colored in yellow and the position of exonuclease insertion in pol-C in red. DNA replication in prokaryotes. Prokaryotic replication is well represented by studying the mechanisms in E. coli. The overall process of replication is segmented into initiation, elongation, and termination. Initiation. Sequential assembly of a large protein complex of ORIC. In E. coli, DNA replication initiates at a short stretch of nucleotides which provides E. coli proteins eukaryotic proteins functions. DNA ORIC proteins recognizes the origin of replication. Gyrase topoisomerase I2 relieves positive supercoils ahead of replication. Fourth DNAB MCM-DNA helicase that unwinds parental duplex DNA CCDC6, CDT1 loads helicase onto DNA. SSBRPA maintains DNA in single-stranded state. G-complex RFC subunits of the DNA polymerase holoenzyme that load the clamp onto the DNA. Toll-free port Pol-D synthesize leading strand E and lagging strand T have proofreading capability. B-clamp PCNA ring-shaped subunit of DNA polymerase holoenzyme that clamps replicating polymerase II. DNA works with Pol-3 and E. coli and Pol-D or E in eukaryotes. Primase Polis synthesizes RNA primers. Polis synthesizes short DNA oligonucleotides as part of RNA-DNA primer. DNA ligase seals Okazaki fragments into a continuous strand. Poli RNase H. Fen1 removes RNA primers. Poli of E. coli also fills the gap with DNA table 3.1 proteins involved in replication in prokaryotes and eukaryotes. Multiple binding sites for replication initiator protein, DNA, and is known as the origin of replication boric. Replication begins with the formation of a large complex at this boric site that employs 6 different proteins DNA A, DNA B, DNA C, HU, gyrase, and SSB. DNA A is the most important as its binding plays a deciding role in whether replication will initiate. DNA A is an ATP binding protein that binds to asymmetric 9BP recognition sequences in ORIC known as DNA boxes 5PTTATNCACA, figure 3.15. Figure 3.15 structure of E. coli ORIC. The origin of replication is 245 BP long, containing 9 BP asymmetric consensus sequences, which provide high affinity recognition sites for DNA ATP and are also called DNA boxes blue. On the left of this stretch lies an AT-rich region containing 313 mer repeats gray. There are overall 11 GATC sites black distributed throughout ORIC, which provide recognition sites for DAM methyl additional binding sites for accessory proteins like IHF and FIS are also present in ORIC, not shown in the diagram. During initiation, it forms a DNA ATP complex, which actively binds to the fully methylated ORIC, figure 3.16. Once initiation ends, ATPase activity of DNA hydrolyzes the bound ATP to ADP and inactivates itself. DNA C interacts with DNA bound to ORIC and helps recruit DNA B. The latter, as mentioned earlier, is an ATP-dependent helicase that binds to single-stranded DNA and further unwinds it. Figure 3.16 initiation of replication at E. coli ORIC. E. coli ORIC sequence contains specific binding sites for DNA A, the key initiator protein of replication. DNA A, complex with ATP and other additional factors, labeled in green, binds to ORIC and stimulate bending of adjacent DNA, forming an open complex. 2 molecules of DNA B helicase, labeled in gray, are then loaded onto each template strand with the help of the clamp loader DNA C and ATP, labeled in black. DNA B helps in the further unwinding of DNA, and SSB proteins, labeled in hatch, subsequently bind to each strand to maintain them in a single-stranded state. Gyrase relieves the resulting stress on the adjacent duplex. Subsequent binding of primase completes the formation of chromosome. As DNA C leaves the chromosome, primers are synthesized, and synthesis of leading and lagging strands begins. Methylated ORIC can only initiate replication. The E. coli ORIC contains 11 sites of GATC sequence that provide target sites for methylation by DAM methyltransferase. This methylation plays an important regulatory role in the activity of ORIC. Before replication, the palindromic GATC sequences are methylated on the adenine residues of each template strand. However, the outcome of each round of replication is 2 daughter strands with normal, non-modified bases. This generates hemimethylated DNA, consisting of a methylated parent strand and an unmethylated daughter strand. Since fully methylated origins can only initiate the replication, this mechanism is essential for differentiating between the parental DNA strands that require to be replicated versus the already replicated hemimethylated DNA strands in the genome. DNA ATP binds to ORIC and forms an open DNA complex. On the right side of ORIC, the 4 9BP consensus sequences provide a high affinity binding site for the DNA ATP complex. This binding is stimulated by the accessory protein dye resulting in a multimer filament wrapped by ORIC DNA. ORIC also has AT-rich 313 mer repeats located on its left side. DNA ATP further binds to these regions forming an open double complex, thereby stabilizing the single-stranded unwound DNA, which is then covered by single-strand binding protein SSB that prevents subsequent reannealing of the strands. The protein who acts as an accessory protein that stimulates the bending of DNA and opens complex formation. DNA B clamps onto DNA with the help of DNA C generally. 2 to 4 monomers of DNA ATP bind at the origin forming an open complex with 2 single-stranded DNA, and after the release of DNA A, 1 molecule of DNA B clamps around each of these strands. This clamping is assisted by the clamp loader DNA C and ATP, thereby yielding the 3' complex. Each such complex is recruited by each of the 2 replication forks in the bidirectional replication in E. coli. A notable point about DNA B is that it is an unwinding helicase and plays no role in melting DNA, which is usually done by initiator proteins, DNA A in this case. DNA B binding to single-stranded DNA is a signal to hydrolyze ATP and release DNA C. DNA B recruits primase to the replication fork. Each helicase is activated by ATP hydrolysis and moves past each other, sliding the 2 strands in 5' - 3' direction. After spanning in GT, 65 nucleotides, each DNA B activates a DNA G primase, one to prime the leading strand, and the other to prime the first Okazaki fragment of the lagging strand. Subsequently, the B subunit of DNA Pol-3 is recruited onto each template strand. This activates the ATPase activity of DNA A, which inactivates itself to prevent further initiation. DNA G primase initiates both leading strand synthesis and Okazaki fragment. As mentioned earlier, the product of DNA G is a special kind of RNA polymerase that initiates a new DNA strands by forming a short stretch of RNA called a primer. This is required because DNA polymerases cannot synthesize DNA de novo. They require a 3' that is provided by the RNA primer, which is then extended by the DNA polymerase in elongation. Usually, the RNA synthesized by the priming reaction is de novo. However, in some special cases, the primer is provided by preformed RNA that binds to the template. This special mechanism is executed in reverse transcription of RNA and observed among retroviruses. Since DNA polymerases extend the primer in 5' - 3' direction only, there is a need for a priming reaction for each Okazaki fragment on the lagging strand as opposed to a single reaction on the leading strand. Therefore, a 10 nucleotides long RNA primer initiates each Okazaki fragment, which is then taken over by DNA polymerase III. In E. coli, there is only one catalytic subunit of DNA polymerase III responsible for synthesizing DNA in both leading and lagging strands and is called the DNA polypeptide. Some bacteria and eukaryotes have multiple replication DNA polymerases. Elongation. DNA Pol-3 elongates both the leading strand and each Okazaki fragment. This stage is marked by 2 molecules of core DNA polymerase III binding to each replication fork, one fork extending the leading strand while the other extending the lagging strand. The core polymerase associated with the leading strand follows the direction of movement of the replication fork, while the one associated with the lagging strand moves in the opposite direction. These 2 core polymerase molecules are joined together by AT subunit dimer. As discussed in the structure of DNA Pol-3, the B clamp is a homodimer of B subunits that tether the catalytic core of the enzyme to the template strands. While on the leading strand, the polymerase complex moves continuously following its B clamp along the direction of the replication fork till the termination site. The replication of the lagging strand occurs discontinuously. Pol-3 adheres to the fork with the help of a B clamp. As elongation of each Okazaki fragment is completed, core polymerase dissociates from the template strand, but remains tethered to the fork with the help of a T subunit dimer. In a simultaneous process, primase binds to the adjacent region of the lagging strand template and synthesizes another RNA primer for the next Okazaki fragment. Subsequently, another B clamp is loaded onto this newly synthesized DNA primer complex, followed by the reassociation of the core polymerase, which then elongates this new fragment. Okazaki fragments are repaired by coli and DNA ligase. As the elongation of each Okazaki fragment completes, the 5' - 3' exonuclease activity of the DNA polymerase cleave off the RNA primer of the previous fragment, leaving a gap between the 2 fragments on the newly synthesized DNA of the lagging strand. This gap is also filled by DNA polymerase activity, which extends nucleotide addition from the 3' end of the next Okazaki fragment, and eventually DNA ligase joins the fragments. This process of RNA displacement with new DNA is similar to Nick translation, where a DNA sequence is usually replaced by a new DNA segment. Termination. Termination in E. coli takes place at specific sites in the DNA called replication termini tear. E. coli has 10 tear sites, tear H, I, E, D, and on one side and tear C, B, F, G, and J on the other, basically 2 clusters of 5 sites, having opposite polarity. Since E. coli replication forks have 2 opposite orientations concerning ORIC, each fork having a particular direction is impeded by a specific cluster, while the fork moving in the opposite direction passes through, but is impeded by the other. Thus, this arrangement of tear sites in the 2 clusters entrap the replication forks moving in either direction in a distance diagonally opposite to ORIC, figure 3.17. It is reported that the tear sites interact with replication termination protein 2s, which is responsible for restricting DNA helicase activity. 2 tear forms a DNA protein complex that acts as a barrier for DNA B helicase activity. This theory is confirmed by studying in vivo interaction of 2 CNAB by yeast 2 hybrid analysis. Figure 3.17 replication termini in E. coli. A typical E. coli chromosome contains an origin of replication and 10 replication termini tear. The tear sites are oriented such that they allow movement of replication fork in one direction, but block their movement in the reverse direction. The 2' gene labeled in blue encoding 2's protein is located downstream of the TER site. Rolling circle replication. In many cases of circular DNA replication, it is seen that the dotted DNA molecules are linear strands containing the repetitive base sequence of the circular DNA, forming a concatemer. These are formed by a special mechanism called rolling circle replication. In these cases, an initiator protein cleaves one strand of the DS circular DNA to expose the 3' end and a 5' terminus. The 3' provides the primer for an extension by DNA pol 3 taking the unixed circular DNA as a template leading strand. As the DNA synthesis continues, the next strand is displaced by multiple SS linear copies of the original DNA, forming a concatemer. These copies are eventually circularized and converted to dsDNA in the usual process of replication. The initiator protein next to terminate the leading strand replication. RNA polymerase and DNA pol 3 then replicate the ssDNA to a double-stranded circle, figure 3.18. DNA polymerase I remove and replace the RNA primer with DNA, and DNA ligase seal the nick to form a new molecule of circular dsDNA. This mechanism of replication was first observed in bacteriophages. It is also seen in RNA and dsDNA viruses, for example, herpes virus. Figure 3.18 mechanism of rolling circle replication. The circular dsDNA is nicked in one strand by the initiator protein, and the 3' end of the nick is elongated as the leading strand. The 5' end of the nick is displaced as the lagging strand, which is nicked and joined by ligase and replicated to dsDNA by Okazaki fragment formation. DNA replication prokaryotes versus eukaryotes. Unlike prokaryotes, where the genome comprises of a small and circular chromosome, and replication begins from a single replication origin, the genome complexity of eukaryotes requires them to comprise multiple origins to allow the formation of multiple replication forks simultaneously at different locations. A typical yeast chromosome consists of approximately 400 origins, while the number ranges between 30,000 and 50,000 in humans. Although eukaryotic replication is also bidirectional, semi-conservative, and semi-discontinuous, the machinery required to carry out each process is different from those discussed in prokaryotic replication. Most importantly, eukaryotic replication requires multiple DNA polymerases. The nuclear DNA is replicated by DNA polymerase A, D, and E, while mitochondrial DNA is replicated by DNA polymerase G. Most of these replicas are large heterotetrameric enzymes with the main subunit usually responsible for catalysis, and the rest are usually for priming or processivity. DNA polymerase B has higher fidelity than the rest, and it also acts in DNA repair activity. All other DNA repair polymerases show higher rates of error and are known as error-prone DNA polymerases. Replication origins in eukaryotes. In eukaryotes, replication origins were first identified in self-replicating plasmids in Saccharomyces cerevisiae as high-frequency transporting elements. These are later termed autonomous replicating sequences, ARS, as they can confer autonomous replication in yeasts. ARS contains 11 BPAT-rich consensus sequences called autonomous consensus sequences, ACS, which interact with the hexameric fork. The origin recognition complex, fork, is highly conserved in all eukaryotes and was first found in S. cerevisiae, often called SWORK. Forks have now been characterized in other eukaryotes, for e.g., Schizosaccharomyces pompa, SWORK, Drosophila, DMORC, and Xenopus X LORC. Adjacent to ACS lies the B domain consisting of B1, B2, and B3 elements, figure 3.19. B1 also interacts with work along with ACS, while B2 binds to the MCM2 to 7 complex. B3 binds to transcription factor ABF1, ARS binding factor 1, which plays an important role in maintaining the chromatin structure and nucleosome assembly. Figure 3.19 Eukaryotic origin of replication ARS of S. cerevisiae. The initiation complex is a large protein complex called WORK, which is shown to bind to the ACS labeled in blue as well as the B1 element. Sequences 5' to ACS are identified as domain C labeled in green, while those 3' to ACS are identified as domain B labeled in gray. The latter is divided into different subdomains viz. B1, B2, and B3 are grouped into sequence-specific elements depending on their functions. Initiation of eukaryotic replication is carried out by DNA polymerase A, which has kinase activity and functions as RNA polymerase to produce RNA primer on both the leading and lagging strands. The enzyme has a 180 kD catalytic subunit responsible for polymerase function and 2 smaller subunits required for kinase activity, and AB subunit necessary for assembly. DNA polymerase A binds to the origin and synthesizes 10 bases long RNA, which is then elongated on the leading strand by DNA pol B and the lagging strand by DNA pol D. Because of its priming activity, DNA polymerase A is also called primase. The MCM helicase, the eukaryotic homolog of DNA B, unwinds the dsDNA in the process to aid in the action of DNA polymerase A. pCNA is a eukaryotic homolog of B clamp that maintains the processivity of DNA polymerase D, and RFC is a homolog of the eukaryotic G clamp loader that helps load pCNA onto the DNA. DNA polymerase D complexes with pCNA, which keeps the enzyme tethered to the replication fork during lagging strand elongation. One striking difference of eukaryotic DNA polymerase from prokaryotic polymerase is that the former lack 5' minus 3' exonuclease activity. Therefore, during the synthesis of Okazaki fragments by DNA polymerase D, the enzyme is unable to remove the RNA primer of the preceding fragment. This job is performed by FEN1, flap endonuclease 1, which has 5' minus 3' exonuclease activity. The elongation of Okazaki fragment removes the RNA primer of the preceding fragment as a flap. This is removed by FEN1. Hence, the activity of this enzyme is analogous to the nick translation performed by DNA polymerase I in prokaryotes. DNA ligase is present in eukaryotes as well, which then seals the nicks between the Okazaki fragments. A typical eukaryotic replication fork is represented in figure 3.20. Figure 3.20 a schematic illustration of the eukaryotic replication fork. The replication fork represents all the components studied in the model eukaryotic virus SV40. The T antigen of the virus exhibits helicase activity, similar to MCM helicases, and pCNA and RFC represent the B clamp and clamp loader, respectively. RPA represents the eukaryotic equivalent of single-strand binding proteins SSB. Pol on the leading strand and Pol on the lagging strand represents the core polymerases required for elongation, while Pol represents primase. FEN1 and ligase are recruited on the lagging strand for processing of Okazaki fragments. Polymerase switching. An important aspect of eukaryotic replication is the coordinated control of all the polymerases and an intricate crosstalk that is reflected in the phenomenon called polymerase switching. As soon as DNA polymerase synthesizes primers on both leading and lagging strands, the template needs to be handed off to polymerase E and D, respectively, for elongation. Polymerase switching refers to this event of handing off the template to one polymerase from another. However, polymerase A is still required for repeated priming of Okazaki fragments, only to be switched to polymerase D for elongation of each fragment. Polymerase switching is very critical in these steps and is usually carried out by interactions of different proteins, i.e., polymerase A, D, proliferating cell nuclear antigen pCNA, replication factor C, RFC, and replication protein A, RPA. Polymerase switching is also seen during repairing Okazaki fragments by FEN1, a DNA ligase that acts together in the association of pCNA. The replication termination in eukaryotes occurs by a highly complex mechanism which is still being elucidated. The most common mechanism studied so far is by ribosome disassembly, which is marked by the unloading of CMG helicase from chromatin. This is usually observed during the late S phase when ubiquitin chains are assembled on MCM7 for full ubiquitination, which is reported to be linked to CMG unloading. Dissociation of both MCM7 and CMG helicase requires E3 ubiquitin ligase dia2. However, several studies suggest that after unloading, MCM7 does not undergo proteasomal degradation. Instead, CMG is recycled after ribosome disassembly. As a consequence of CMG unloading, all other proteins, including pol B, are disassembled from the DNA. pCNA is reportedly unloaded by L1, leading to the removal of FEN1, ligase and pol B involved in lagging strand elongation, figure 3.21 and figure 3.22. Figure 3.21 the DNA replication cycle and the centrosome duplication cycle. The DNA replication cycle is shown inside the circle, while the centrosome replication cycle is shown outside the circle. The proteins with double arrows denote their function in coordinating both cycles. Inside the circle. At the late M and G1 phases, ORC, CDC6, and CDT1 recruit MCM helicases to the replication origins to form a pre-replication complex, pre-RC. Once the pre-replication is assembled, the origin is licensed to replicate. Upon entry into the S phase, CDC45 and GINs are recruited to the replication origins dependent on DPP11, SLD2, and SLD3 under the regulations of CDK2, cyclin E and CDC7 kinases. The phosphorylated MCM2 to 7 helicase, together with CDC45 and GINs, forms a CMG complex and functions to unwind the DNA replication origin site. Subsequently, pol E and pol D are recruited to the replication fork, and DNA replication initiates. The replicated DNA duplexes are held together by cohesin in the G2 phase and separate in mitosis, outside the circle. During the G1S transition in the same cell cycle, centrosome duplication initiates from centriole disengagement and takes place during the S phase. CDK2, cyclin E activity is required for centrosome duplication initiation. The replication licensing factors ORC1, gemin and maybe the others function by preventing the centrosome reduplication after centrosome duplication initiation. Centriole elongation and maturation occur during the S and G2 phases. At the G2M phase, the duplicated centrosomes are separated to form the poles of the bipolar spindles to execute mitosis. Figure 3.22 assembly of the pre-initiation complex, pre-IC. This figure reveals how the pre-initiation complex, pre-IC, during DNA replication initiation is assembled and shows that pre-IC assembly separates the core component of the replicative helicase, the MCM2 to 7 double hexamer. All firing factors associate with replication origins to assemble pre-IC before MCM10 functions. Pre-IC assembly is mutually dependent on all the firing factors whose association with replication origins is triggered by CDK. In the pre-IC, not only the formation of type CDC45-MCM2 7-gens CMG complex, but also the separation of MCM2 to 7 double hexamer occurs. There are also events where replication termination appears to be site-specific. One class of site-specific replication is by replication fork barriers RFB. The process of termination takes place by the convergence of two replication forks coming from two directions of particular sites in the genome called RFB, which are bound by RFB proteins installed in replication forks. For example, the ribosomal DNA locus in button yeast contains a termination element, TER, that binds a terminator protein fork blocking protein 1, FOB1 forming a similar complex like the Tuster complex in E. coli replication termination. The second type of site-specific termination event occurs at telomeres. Since eukaryotic chromosomes are linear, once the replication fork reaches the chromosomal end, CMG disembarks itself from the leading strand template so that pole reaches the end of the chromosome. On the lagging strand, CMG tethers pole to the ribosome, and as it detaches itself, removes pole in the process. Hence, priming of a new Okazaki fragment cannot be initiated any further. However, this leaves an ssDNA region on the 3' end of the template strand, which is not replicated. Since replication is bidirectional, each parent strand encounters this problem at each chromosomal end. This results in progressive shortening of the daughter chromosomes with each round of replication. This is known as the end replication problem. Concept of telomerase. The end replication problem in eukaryotes is tackled by a specialized DNA polymerase called telomerase. It contains an RNA component that contains 1.5 copies of the telomerase sequence and a reverse transcriptase enzyme called telomerase reverse transcriptase or TERT. The RNA anneals to the unreplicated ssDNA of the 3' end of the template strand. TERT then synthesizes DNA complementary to the RNA template, and with repetition of this process, the 3' end of the parent strand is extended, providing template DNA for further extension of the 5' end of the lagging strand by lagging strand synthesis. Figure 3.23 and figure 3.24. Figure 3.23 telomere extension of eukaryotic chromosomes by telomerase. The telomerase binds to the 3' end of the telomere and provides an RNA template labeled in blue for the extension of the 3' end. As telomere extends to the length of the RNA, telomerase shifts and repositions itself to the new 3' end, and the extension is repeated. The extended length of the telomere acts as a template for the synthesis of new Okazaki fragments aided by primase and DNA polymerase. Although the resulting DNA still has a 3' single-stranded overhang, both strands of the chromosome are extended in the process, which prevents the progressive shortening of chromosomes. Figure 3.24 classical versus new view of telomere structure. The classical view. The classical view of telomere structure. Linear double-stranded telomere repeats make up the bulk of the telomere sequence. Duplex telomere binding proteins such as RAP1P in yeast and TRF1 in mammalian cells bind along the length of the telomere repeat tracks. At the end, there is a 3' overhang on the G strand. The end is thought to be bound by an N-specific telomere protein exemplified by the oxytricha AB telomere binding protein. The new view. The new view of telomere structure. The telomere DNA loops back on itself, forming a lariat structure. The 3' G strand extension invades the duplex telomeric repeats and forms AD loop displacement loop. Duplex DNA telomere binding proteins bind along the length of the telomere repeats as above. A specialized telomere binding protein binds the D loop at the junction of the lariat. TRF may play this role in stabilizing or allowing the formation of the D loops. Drug targeting DNA replication. DNA replication machinery comprises many proteins, some of which play primary roles, and others are involved in regulatory functions. Nonetheless, each of them is indispensable for the process to function normally. This opens a vast scope of drug design, as any inhibitory molecule targeted to replication protein can stop the replication. Inhibiting replication is mainly important for cancer cells and many other pathogenic organisms, for which the replication machinery is already explored. There are a large number of ways to inhibit replication. 1. Inducing DNA damage by supplementing drugs like cisplatin. 2. By making the replication polymerases dislodge from the replication fork, leaving large stretches of ssDNA exposed, which are subsequently bound by REPA, ultimately leading to the collapse of the replication fork. 3. Use of nucleotide analogs, which are being used for the treatment of autoimmune disorders. 4. DNA polymerase inhibitors have been used to target mitochondrial DNA polymerase to inhibit mitochondrial DNA replication and induce tumor cell death. The subunit of bacterial pol-3 is a potential target for drug design. The first inhibitor design was H-Cura as early as 1970, which belongs to the class of pol-3 inhibitors. In gram-positive bacteria, the pol-3 subunit homolog is called pulp, and most of the drugs that inhibit pol-3 are designed against pulp, while in the case of gram-negative bacteria, the homolog is called DNA. 324C is one such compound targeting pulp, which was reportedly active against B. subtilis in vitro. Pol-3 inhibitors belong to 3 major classes: I, the 6 analymer cells, ALS, E, the guanine inhibitors, and E, the non-nucleobase inhibitors. A class of pol-3 inhibitors competitively inhibit pol-C, and the inhibitory mechanism lies in their binding to the cysteine bases of DNA 6-dehydroxyphenylazote uracil. H-Cura is an example of a no inhibitor. 2 other ALS, 6,3-ethyl-4-methylamino uracil, EMAU, and 6,3,4-triethylene uracil, TMAU, were found to be promising against pulp in vitro, against various gram-positive bacteria, including MRSA. B. The guanine class of inhibitors act by the same mechanism as O, but they have a purine base instead of uracil and competitively inhibit both pulp and DNA. Examples of guanine inhibitors are N2,3,4-dichlorobenzyl guanines, DCBGs, and the N2,3-ethylphenol guanines, EMPGs, which have shown potential antimicrobial activity against several gram-positive pathogens in vitro. C. The non-nucleobases that act as DNA polymerase 3 inhibitors include aniline pyrimidinones, APs, and quinazolin 2-YLAMINOQUINAZOLIN4-ols bisphenols. The APs are structural isomers that also competitively inhibit guanine nucleotides. Unlike ALS and APs, bisphenols exhibit a different mechanism by competing with the DNA template instead of nucleotides. 5. Inhibitors of sliding clamps are used for their anti-cancer and antibacterial activities. The sliding clamp is responsible for the processivity of DNA pole, and the B subunit is targeted by drugs like bristlemycin isolated from the bacterium Streptomyces riceus, a natural product of Streptomyces riceus. It is active against mycobacterium species. A modified version of this drug, cyclohexyl bristlemycin, was seen to be highly effective in killing M. tuberculosis in vitro. 6. Helicase inhibitors are also used as drugs for the treatment of cancer. 7. Ligase inhibitors act as competitive inhibitors, and 2 major classes of ligase inhibitors have given the most effective results, viz. 2-amino-1, 8-naphthyridine 3-carboxymides, ANCs, and adenosine analogs. ANCs are mainly active against Liga gram-positive pathogens like MRSA and C. difficile. Adenosine analogs are effective against both gram-positive as well as gram-negative bacteria and exhibited promising results in Staphylococcus aureus and Streptococcus pneumoniae infections in animal models. However, the prospect of developing resistance to these drugs due to single-base mutations has limited the entry of this class into human trials. 8. Topoisomerase inhibitors fall into 2 classes of aminopurines, which are ectase inhibitors, and fluoroquinolones, which interfere with the catalytic activity of topoisomerase 2. Novobiocin is an example of the first class, which has been licensed for the treatment of staphylococci infections. Novel non-quinolone topo 2 inhibitors, NBTI, have also been developed, which also target the catalytic core of topo 2 but exhibit a different mode of action. Against mycobacterial infections, it is to be noted that of all the essential replication proteins, the fluoroquinolone compounds most effectively target DNA gyrase, which is used in the treatment of MDR-TB, telomerase, and cancer. Upregulation of the TERT gene encoding telomerase enzyme is a key to the highly proliferating ability of all cancer cells. Telomerase is a ribonucleoprotein complex containing reverse transcriptase activity and a functional RNA component encoded by HTR, also called hTERT. Inevitably, this enzyme provides a promising target for anti-cancer drug development. One such intravenous drug is imetelstat, which competitively inhibits this enzyme and was developed for the intravenous treatment of many cancers. It consists of a consensus oligonucleotide sequence 5' palmitate tag NH2-3' that is complementary to a 13-mer region of HTR at the active site of telomerase, resulting in complete inhibition of the activity of the enzyme. The inhibitory action of the drug has been demonstrated as a shortening of telomeres in various cancer cell lines, viz. bladder, breast, lung, liver, prostate, and pancreas. As telomerase of cancer cells undergoes degradation by proteasomes, the resulting peptides are presented on the cell surface as antigens. Interestingly, these antigenic epitopes can be recognized by cytotoxic T cells, and tumor cells can be destroyed by eliciting an immune response. This strategy is exploited in developing telomerase-based immunotherapy approaches, which have already been effectively used in cancer patients. 3 hTERT vaccines that have been successfully developed so far are GB1001, VX001, and GRINDAC1. Synthesis of the lagging strand requires a short primer, which will be removed. At the extreme end of the chromosome, there is no way to synthesize this region when the last primer is removed. Therefore, the lagging strand is always shorter than its template by at least the length of the primer. This is the so-called end replication problem, figure 3.25 and figure 3.26. Bacteria do not have the end replication problem because its DNA is circular. In eukaryotes, the chromosome ends are called telomeres, which have at least 2 functions. 1. To protect chromosomes from fusing with each other. 2. To solve the end replication problem. Figure 3.25 telomerase and telomere extension. To extend the length of the telomerase first extends its longer strand. Then, using the same mechanism as synthesizing the lagging strand, the shorter strand is extended. Figure 3.26 the mechanism of telomere extension by telomerase. In a human chromosome, the telomere is about 10 to 15 kilobits in length, composed of the tandem repeat sequence, TTAGGG. The telomerase contains an essential RNA component which is complementary to the telomere repeat sequence. Hence, the internal RNA can serve as a template for synthesizing DNA. Through telomerase translocation, a telomere may be extended by many repeats. In the absence of telomerase, the telomere will become shorter after each cell division. When it reaches a certain length, the cell may cease to divide and die. Therefore, telomerase plays a critical role in the aging process. DNA repair. DNA repair. Despite the proofreading functions of DNA polymerases, there are often errors during replication as a result of the misincorporation of nucleotides that are retained in the DNA sequences. Cells have additional mechanisms to cope with such errors. Such repair mechanisms usually work in two fashions. Some are general repair mechanisms, others repair specific types of damaged DNA. Mismatch repair. This system of repair enzyme is called mismatch correction enzyme and is encoded by MUF, MUL, MUTS, and MUT2 genes. The enzyme recognizes any incorrect residue left in the daughter strand during replication and excises the residue leaving a gap which is eventually filled by DNA pole with the incorporation of the correct nucleotide. The process by which the mismatch correction enzyme discriminates between parent and daughter strand is interesting. Immediately after replication, the newly synthesized daughter strands pass through a lag period before they are methylated by DAM methylases. During this period, the parent strands are all methylated, thus giving a chance for the mismatch repair system to operate on the unmethylated daughter strand. Figure 3.27. The MMR system in E. coli is essentially composed of four identifiable proteins: MUTS, MUL, MUF, and UBRs. MUTS. MUTS is the pivotal protein of the MMR system that can detect mismatches in the double-stranded DNA. It initiates the MMR machinery by recognizing non-specific interactions. It has two functional domains, including a DNA-binding domain and an ATPase dimerization domain. Both domains of the MUTS protein are sterically distant in the inactive state, but conformation changes on recognizing a specific lesion in DNA, ATP, or just binding to DNA. Both protein domains affect each other's function. MUL. MUL protein acts as a mediator between the MUTS and other protein complexes in the MMR systems. It creates a link between the MUTS mismatch recombination and the excision of the mismatch bases by responsible proteins. MUL interacts with the activated MUTS homodimer and sequentially stimulates the endonuclease activity of the MUF protein. In addition, MUL can also function as a recruiter of UBRD, which facilitates its loading onto the DNA lesion site. MUF. MUF is a member of the type 2 family of restriction endonucleases. It cleaves the mismatch-containing DNA single strand, specifically at hemimethylated GATC sites. MUF knitting activity is stimulated by the MMR complex consisting of MUTS, MUL, and ATP. The C-terminal helix of MUF protein acts as a molecular attaché, through which MUTS and MUL communicate and activate MUF. UBRD. UBRD is a DNA helicase tube that unwinds DNA starting from the nick generated by MUF. It is loaded onto MUL at the DNA duplex lesion site, which increases its intrinsic helicase activity. The nick generated by MUF serves as a point of entry for single-stranded DNA binding protein and UBRD helicase 2. The loading of UBRD helicase 2 at the nick is facilitated through protein-protein interactions with MUL. In addition, many other proteins also take part in this process, including free single-strand DNA-specific exonucleases in MMR system. The 3'-5' exonucleases XY and exoxin, the 5'-3' exonucleases, which are responsible for the excision of the newly synthesized DNA strand between the nick and the mismatch. Figure 3.27 methyl-directed DNA mismatch repair. Excision repair. This repair system comprises a battery of endonucleases, each acting on a specific type of DNA lesion. For example, the most common lesion is caused by deturination, where the sugar phosphate backbone is left intact with a missing base. AP endonuclease recognizes such exposed sugar and excises the altered sugar by hydrolyzing the phosphodiester bond on its 5' side. This results in a gap in the DNA that is filled in by DNA polymerase and ligase activities. The above repair mechanisms act on single-stranded breaks SSB on DNA, hence known as SSBR. In base excision repair, BER, specific DNA glycosylase act on altered bases in the DNA and excise them. For example, removal of deaminated Cs by uracil glycosylase. Such base excision mechanism gives rise to exposed deoxyribose sugars with a missing base. These sugars are then recognized and acted on by specific AP endonuclease, similarly as discussed above. Another type of excision repair is nucleotide excision repair, NER, where the repair system specifically removes any kind of bulky lesions. These are usually formed by covalent reactions. For example, pyrimidine dimers that are formed due to exposure to sunlight. UBRBC nuclease scans the DNA strand for such lesions, and once found excises them by cleaving the phosphodiester backbone on both sides of the lesion. The portion of the strand containing the lesion is removed by the action of the DNA helicase enzyme, leaving a gap of around 12 nucleotides which is eventually filled by DNA polymerase and joined by ligase. Impaired NER has resulted in a rare genetic recessive disorder in humans, called xeroderma pigmentosa, where cells are unable to remove DNA lesions due to damage by UV. Patients with such disorder exhibit increased sensitivity to sunlight, having a high risk of developing skin cancer. Cockayne syndrome (CS) and trichotillomania (TTD) are human diseases that arise from defects in the NER pathway. Figure 3.28 DNA-based excision repair pathway. Photoreactivation. This mechanism can also remove thymine dimers by directly breaking the covalent bond between the thymine bases. Such type of activity is exhibited by the photoreactivation enzyme 3. This type of repair system does not require the activity of DNA polymerase and ligase as it directly breaks the bonds forming the dimer utilizing energy by absorbing photons of sunlight. That is why this type of mechanism takes place only in the presence of light. Figure 3.29. Figure 3.29 Photoreactivation repair DNA versus base excision repair. Double-stranded break DSB repairs we have seen earlier in mismatch and base excision repair. Any damage in the daughter strand can be repaired by replacing the correct nucleotide complementary to the template strand sequence, which is easily done by DNA polymerases. However, often the problem arises where a lesion or a base pair damage has left a gap on both the strands. The sequence on the template is no longer available for any polymerase to act on. In the mammalian system, two major pathways exist to repair this kind of damage: homologous recombination (HR) and non-homologous end joining (NHEJ). In the former pathway, a recombination repair protein nicks a piece of template DNA from a homologous chromosome which is used as a template to fill the gap on the damaged DNA by recombination. NHEJ pathway, on the other hand, employs a complex of proteins that bind to the cleaved ends of the DNA and initiate a cascade of reactions to join the break. The DSB is recognized by a heterodimeric Q protein that binds to the break and recruits a DNA-dependent kinase DNA PKCs. They bring the ends of DNA together, which are then acted by DNA ligase IB to seal the break.